Ladies and gentlemen, now we will proceed to the presentation section of the theme-based research projects under the theme Promoting Good Health. Each presentation will last for 35 minutes, followed by a 10-minute Q&A section. Today, we are very pleased to have Professor Kwok Wai Law to be our facilitator for this section. Professor Law is the professor of the Department of Anatomical and Cellular Pathology at the Chinese University of Hong Kong. Professor Law is specialized in molecular genetics of nasopharyngeal carcinoma, NPC. He is also the project coordinator of the theme-based research scheme-supported project. May I now hand over the stage to Professor Law, please.
Thank you very much for your kind introduction. I'm Kwok Wai Law from the Chinese University. So here I would like to introduce the first theme-based project presented by my colleague, Professor Nathalie Wong from the Chinese University of Hong Kong. Actually, the project title is the Liver Cancer Genome Project and Translating Genetic Discovery to Clinical Benefits. In this project, the team will aims to use the next generation sequencing technology to dissect the molecular base of the liver cancer and aims to develop the new diagnostic marker or new therapeutic approach for this cancer. So Professor Nathalie Wong, please come to the stage and deliver your talk.
So thanks very much, Kwok Wai, for your introduction. So I'm Nathalie Wong, speaking on behalf of the group of the theme-based research project on the liver cancer genome project. All right. So this project, in fact, I want to say it's a joint effort of a couple of institutes in Hong Kong, Hong Kong UST, CUHK, Hong Kong U, and also the Sun Yat-sen in Guangzhou University, as well as BGI from Shenzhen for their assistance in the sequencing work that we have done. So before I go into the subject of HCC, perhaps I'll just give a couple of slides on the background of cancer, and also in Hong Kong. So the first thing is that cancer is a major public health problem, not only in Hong Kong, but also worldwide. And it affects all people, irrespective to their age, gender, and also the social economic background. In Hong Kong, cancer remains a major cause of our deaths and also of our healthcare system. So if you look at this chart of the top 10 cancers in Hong Kong in the year 2015, you'll see that the first rank is colorectal cancer, liver comes somewhere here at number 5. But however, this is the incidence. But however, if you look at mortality, liver cancer is actually the third. And the reason being is because the incidence to mortality ratio is high because the treatment of liver cancer has actually it's actually not very good, it's fallen behind. And there's a couple of reasons as well, so I shall highlight in a minute. So hepatocellular carcinoma, in short HCC, actually accounts for more than 90% of all primary liver cancer. So very often we use the terms very loosely. Hepatocellular carcinoma refers to liver cancer. It is the third leading cause of cancer deaths in Hong Kong, as I showed you earlier on in the slide, and the second in mainland China. An annual incidence of about 320,000 has been reported per year, of which more than 50% actually occur in China. So this is a disease that is highly relevant to us. The low therapeutic efficacy in the majority of patients actually produces those poor survival prognosis. Sorafenib is the first FDA-approved drug for HCC. However, despite the fact that the Western data shows an 11-month median survival, in Chinese or Asian populations, the median survival is only 6.5 months, and the placebo is only 4.2, so it's not much benefit. There are several reasons in addition to the low therapeutic efficacy. That's why the disease carries such poor prognosis. The limitations in early detection, i.e. the HCC surveillance methods, also has got its limitations. At the moment, we're still using serum alpha-fetoprotein as one of the markers for it. However, we know AFP, or in short for alpha-fetoprotein, has got a sensitivity of around 70%. There's associated with false positivity if the patient has got active hepatitis. And worse all is the false negativity. About 50% of the early HCCs could be missed. Another way of surveilling for HCC would be the ultrasound scan. However, it's labor-intensive, operator-dependent. Again, the sensitivity is only about 80%. So we do have some limitations in the management of HCC. So we all know cancer is a genetic disease arising from the defects of the genome, and there is no difference for HCC as well. Liver cancer is also a genetic disease. Hence, we believe as a group that understanding the genetic background of HCC is key to the better development of anti-cancer therapy treatments. This project hence aimed to improve the clinical outcome of patients with HCC by clarifying the multifaceted genomic alterations that underscores the biology of HCC. If we look into the etiology of HCCs, there are quite a few of them. In the States, up and coming, there are lots of the metabolic syndrome associated with HCC arising from obesity and diabetes. Places like in Egypt, you will have a high incidence of hepatitis C, as well in Japan. In places like in Africa, like Sudan, the aflatoxin exposure for the poorly stored grains, that will induce also a higher incidence of HCC. Here we are in China, we have a high incidence of hepatitis B infection, and that accounts for more than 80% of our cases. And also Mongolia, it will be Hep C, Hep B, and alcohol. Back here at home in Hong Kong, hepatitis B, as I mentioned, is one of the strongest etiological factor for us. And it is known that chronic carriers of hepatitis B actually will have 100 to 200 times more risk of developing HCC compared to those non-carriers. More recently, here in Hong Kong, there is an increasing prevalence of the non-alcoholic fatty liver, in short called NAFLD, and this itself can also produce a certain amount of risk for further development to HCC. The progressive change from NAFLD to NASH, which is the non-alcoholic steatohepatitis, and subsequent development to cirrhosis is believed to be a high risk for development of the eventual cancer. So hepatitis B, at the moment worldwide, we have 400 million infected individual carriers of it. This is considered a high-risk population, of which 100 million people will have the active hepatitis, gradually develop into chronic hepatitis, and each year is believed about 4% of these individuals who are from chronic hepatitis B infection and cirrhosis would develop into HCC. And as I mentioned earlier, it's about 320,000 per year, and most of them here in China. The epidemiology, as I mentioned, is evolving, and very much is due to the metabolic syndrome, where the hyperlipidemia, fatty liver, type 2 diabetes, all of which, and hypertension, all of which contributing to the obesity, actually becoming increasingly recognized as a major risk factor. So under metabolic syndrome, which is an emerging cause of HCC, we have at the moment about 700 million of NAFLD individuals. Of this high-risk population, about 200,000 of them will have NASH, and then some of them will develop into cirrhosis, and about 3% per year will develop into HCC. At the moment, we are seeing 50,000 cases per year, but the trend is increasing. And NAFLD is an important public health problem in the 21st century and could be very important for us here in China. So we have two major etiological factors in Hong Kong, and we know that both of them can induce liver cirrhosis. And at some point, this liver cirrhosis would transform into HCC, and this is where this project team has spent 5 years trying to investigate why does it change into HCC. And so we turn to the genetics. Surrounding the central dogma of carcinogenesis is that we know that within the normal tissue or normal cell, there will be some reason, being the etiology, being the viral infection, whatever, can impinge on the genome and cause a mutation that may confer certain growth advantage to the cell. From here, the cell will proliferate, not going through the programmed cell death, and at some point may introduce another mutation through the active proliferation, through the regenerations and all that. As a result, this may cause the first primary tumor, which can proliferate and increase the number of mutations as it replicates, and then gradually may even develop into a metastasis-initiating clone. So mutations is a very important thing for us to understand and try to elucidate the genome, especially in terms of understanding carcinogenesis. But we know that within our genome, we have 3.2 billion nucleotides of these ATCG. It's a lot to elucidate. And we know that these ATCG translates to about 20,000 coding genes or coding proteins. So we then, to really elucidate these genomes for each and every single patient that we have analyzed, we need to use the state-of-art technology at the time. The state-of-art technology at the time was the massive parallel sequencing, or some of us will call it next-generation sequencing, or in short, NGS. So I will not go too much into the detail of the technology, but just 2 slides, very straightforward, just to highlight to you what we have done. So in this flow cell, we will actually introduce the DNA template from the whole genome of the patient. And from here, they will actually replicate by itself, okay, forming clusters. And then the sequencing will go up and go down, giving us two ends of the sequencing data. And the sequencing is done by the sequencing by synthesis, where the fluorescent nucleotides of the ATCG will be introduced as they try to read the sequence on the template. And from there, we'll have the information of the sequencing. So here is what I mean, the ATCG, each of them in different color. The cluster is in black, and so at each level, it represents a nucleotide incorporation. And like that, the TCGA is being read. So with this one flow cell information, what we would get, not only the sequencing data, raw sequencing data coming out from a machine, but through extensive bioinformatic analysis, we literally can get a lot of information from one sequencing result. That includes the mutation, which I just mentioned about, the copy number variation, including the deletion of a genome or amplification of a genome, and structural variations, such as translocations. What more we can do to the same patient's tumor is to take the RNA, not only just the DNA, but the RNA, and do the same sequencing work. And what we'll get is the transcript or gene expression level, those 20,000 genes expression level of up and down. We can also have that as well. So, and plus, because we have a clinical team on board, so we will have also the clinical data of the patient, and we can have a really comprehensive information per case, per sample, on what's happening to the genome of that tumor. So based on the genome sequencing of the whole genome sequencing, we actually will be able to first get the mutational pattern. And so this is what we got from the hepatitis B, a set of mutational pattern of the trinucleotide, because trinucleotide, 3 nucleotides form one amino acid. So we want to know the amino acid status. So we have the Hep B related HCC and the NASH related HCC. And then what you could see straight away is that in both of them, there is a transition. So the C nucleotide mutated to the T nucleotide or vice versa. So this is called transition. And we know that based on the breast cancer story, a lot of transition is seen there, and that's really due to the ApoPAC family of enzymes being defective, and we are seeing that in HCC as well. Perhaps really interesting is here. In the NASH group, we are seeing a high transversion. Transversion is a T to A or A to T, where we're seeing a bit more high on the transversion here in the NASH group than in the Hep B group. And it is known that now that the transversion from the A to T or T to A is a mutational signature of the aristolochic acid. In Chinese, we call it Ma Dao Ling Xun, or in short, in English, would be AA, which is a natural product in the Chinese herbal remedies. This remedy is actually for weight loss, for relief of menstrual pain in ladies, and for treatment of rheumatoid. So we are seeing the signature in here in the NASH group more so than in the Hep B group. And this is how it looks like as a dried product. Perhaps surprisingly, is a recent paper that I have to bring into this talk to highlight a point, is that 2 months ago, there was this paper in Science Translational Medicine, which also did this mutation analysis, but in a much bigger database, a much bigger size. And what they found was also a signature of this AA. Perhaps even, and this suggests that this really highlights a different degree of AA exposure, or the AA signature, low, mid, and high, that sort of. Well, perhaps is that they then went on and analyzed a lot of cases from the Southeast Asia, and what they found is a very profound AA signature in many, many of the HCC tumors there, including Japan, Korea, Vietnam, China, and so on. Why is it important, this signature AA? That's because the AA signature is associated with a higher mutational load. So anywhere here that you see in red is the AA signature, and here gives you the number of mutations. So the exposure to AA, this acid, actually correlates quite strongly with the increased mutational burden within the tumor. So it does suggest that some of the herbal remedy that we are having may not be so good to us. So I think, and I agree what they concluded in this paper, is that some measures may be required to avoid the AA exposure in some of the herbal remedy mix so that we can minimize the risk of the development of the cancer. The next thing what I want to go into is based on the genomic blueprint that we have generated on HCC, in fact, we can also define those important driver events that causes the transformation as we were trying to investigate for. We've analyzed 103 HBB, and we analyzed 100 NAFLD NASH HCC, and really this is just an overview of our finding. So what we found is that a couple of major pathways that is actually driving the two carcinogenesis. So each of these lines actually means that some sort of aberrations. The code is over here. So the Wnt signaling mainly is driven through the beta-catenin and auxin-1, where we do see much higher in the NASH-related group than the Hep B group, and vice versa for the auxin. These are two major players in the Wnt pathway. TP53 major really is driving the cancer in the Hep B group in our locality, about 30% in the NASH group. CUHK promoter is also high in both. And then also the chromatin, which is really related to the epigenetics regulation. Notch signaling is very interesting because we're seeing lower of that in the Hep B, but seems to be more in the NAFLD NASH group, which may be suggesting that besides those pathways that are shared between the two, there could be one which is more prominent in the other. As I said, one genome sequencing results can give us a lot of information. It can also give us information on the copy number variation. And what do I mean? So this is the genome copy number variation of one case throughout its whole genome. So if I was to draw a line across, so that's balance. Anything above the line is gain, anything below the line is lost. So you can see here there are some gains and losses in the genome. But because we did, when we do the whole genome sequencing, we will have a reference as well. So that invariably gives us more information. And what do I mean by that? So we can generate what we call the biallelic fraction. Okay. I understand some of the audience here may not be in the genome field, so what I'll do, I'll explain a little bit more. So within our body, one copy is from our father, the parental, and one copy of our genome is from the mother, so we should have two. Anytime when the two copies are present in more or less the equal amount, what we should see is baseline. Two copies very close to each other. However, when there are some copy losses or copy changes, then the difference between the two gets bigger and bigger. So this information is very important for us doing genome analysis. Because if I look at this chromosome 1q and I look at this one, it just tells me one copy of whether the paternal or the maternal has replicated itself. If I look at the 6q, which is a lost, and the gap is big, which means that one copy is lost. It's very important in areas like this. Chromosome 3, the line is very solid on the baseline. If I was just to look at this data, it would tell me that there's nothing wrong with chromosome 3. But if I turn to the biallelic fraction, there is a big gap there, which then tells us one copy is lost, the other copy replicates itself and go back to balance. So like that, we can generate a lot of information based on the genome sequencing and bioinformatic analysis. This case has got still also the relapsed tumor. Again, we can see the genomic pattern based on the total and based on the biallelic fraction ratio. Perhaps even more interesting is that we can look at the gap difference. So the primary tumor, if you look at this chromosome 22 or 21, the gap difference is very minimal. Whereas if you look at the relapsed tumor, the gap difference is very big, which suggests to us that there is a clonal enrichment for aberrations on chromosome 21 in the relapsed tumor compared to the primary. So like that, we generate data and understanding the actual mechanism behind the copy number changes. And this really gives you just a very quick overview on the genome-wide copy gains and copy loss. Areas that where blue represents lost, areas where it's actually reddish represents gains, and the different shades really means degree of gains or loss. So when we compare the two types, I'll just highlight for you, we're seeing that there is a higher incidence of 6p gain in the NASH group and also in the 20q13. So already, by doing this kind of work, it's giving us directions, homing to areas which may be important in driving one cancer than the other. There are also some things that are prevalent in the Hep B. Informatic analysis is indispensable in doing this kind of NGS or sequencing work. We need to work hand in hand with bioinformaticians. So using some of the informatic analysis, what we can do is that we can start integrating copy number change with mutations. And this kind of integration can straight away highlight to us what are the major drivers in terms of the carcinogenesis. So here it shows you the Hep B group and the NASH group. Well, you can see that in the Hep B group, TP53 and its related pathway is a major driver. And beta-catenin is somewhere tucked in here. Whereas in the NASH group, it's a switch. Beta-catenin come out, TP53 gone in. So based on this analysis, what we think is that HCC, in respect to its etiology, at least in NASH and Hep B, it's the same kind of pathway being involved. But there's a switch. It's a switching partner, where TP53 pathway will have a bigger role in Hep B, whereas beta-catenin Wnt signaling will have a bigger role in the NASH pathway. So like that, we have been able to define critical pathway related to the two etiologies as well. So I just really want to highlight a little bit more on why mutations are important and also bring in the beta-catenin story. So if we then home into beta-catenin and ask the mutations where they are, almost invariably, they will be on the exon 3 GSK3 beta phosphorylation domain here. And this data actually is generated in addition to ours. Also, this 88 cases actually from Hong Kong U, and then the French group from the 242 cases. So you can see straight away, not only it's clustered into this region, but also there are mutational hotspots, S45P and C41A. We then also did some cloning of each and every single mutations that we picked up. And then what we saw is that these mutations actually hold functional importance. Each mutation will actually cause an increased growth, as you can see here. What the group actually is currently working on, because Wnt signaling is a very important physiological pathway, so you really can't use the common cytotoxic drug to try to attack it, because you will also attack the normal cells as well. So what we're doing now is actually a more focused approach, is that we know that beta-catenin, because of this mutation, will have an increased chance of going into the nucleus, and normally what beta-catenin will be binding will be to the CBP and the transcriptional factor TCF. But it is possible now, through our further research, that there is a Wnt modulator that can actually affect the binding between the two. As a result of inhibiting the binding between beta-catenin, the co-activator, and the CBP, what we invariably do is to stop the active proliferation, because these genes are involved in active cell cycle and proliferation of cell. What we then did was then to push the partner of the beta-catenin to P300, and another subset of gene is being produced, which is involved in differentiation. So in a way, by modulating the partner with the beta-catenin, we actually can bring down the speed of growth and then to push the cell into differentiation, and will slow down the growth as such. And then that would open up another option of combinatorial therapy in terms of targeting HCC. We, in fact, did quite a bit of work on trying to understand the etiological genetic associations. I will highlight a little bit more. So here is, we understand from that the pathology of for NAFLD and NASH actually followed a specific steps from healthy liver, steatosis, NAFLD, and NASH, and fibrosis and HCC. And to understand also what's being transcribed in these different pathological features, we actually use transcriptome, which is RNA-seq. And also we use animal models so that we can have a better idea to follow the natural course of the disease, because very often we will not be able to get our hands on these tissues of some of the early stages, so we have to depend on animal models. And also, we have to still work on the patient samples and also to use these tissues in parallel with the genome sequencing work and do integrative analysis. So here shows one of the interesting genes that we picked up. This is the OGT, which is in the NAFLD-related HCC. We were able to substantiate our finding in two cell lines that we have established from the group, basically from the NASH NAFLD patients that we have established, and we used the same cell lines to actually define its functionality. And we were able to define the importance of the OGT in the development of the ER stress, inflammation, and proliferation. Perhaps the most interesting part would be the targeting aspect, where through understanding the pathway, defining it, the important players in this under the OGT, that we were able to pick up that by targeting the fatty acid synthase inhibitor, you can actually block this pathway, and of course the specific inhibitor against the OGT can also block the pathway and then the carcinogenesis process. As I said, the animal models we use quite a bit in our studies. So here shows whether wild-type mice was fed with a high-fat diet, the kind of tumor that you generate from a genetic defect, mice is actually very similar. And again, we found some genetic mutations and also the pathways that are involved. Thus, our work actually in the last few years actually reinforces some of the earlier thinking about the NASH-related carcinogenesis. Particularly, we enforce on two paths, the JNK path and then NF-kappa B path in the induction of the NASH-related liver carcinogenesis. Next, I wish to go into the HCC of the Hep B, sorry. So in Hong Kong, as I mentioned earlier, there are more than 80% of the HCC arising from the chronic Hep B infection. We understand that the Hep B infection can exhibit in two forms in the liver genome. One is the CCC DNA, which is the covalently circled DNA, which replicates by itself in the host cell. And the other one is by viral integration. Because it's a DNA virus, it almost invariably will integrate into the host genome. So for many years, people have been postulating or speculating that the integration itself actually is carcinogenesis. But the data was speculative then, but until now with the genome sequencing, we really can nail these questions. So there were two questions in the field that actually is asking about HBV integration. So the first one is, from the integration, does it result in read-through chimeric transcript, i.e., by integrating, does it come out with a bit of the human and a bit of the virus, or is just the virus itself? The second question is that where it integrates, does it affect the downstream gene or the upstream genes in expression? So these are two important questions of the field, and we were able to address this by using sequencing work. So here shows you some of one figure of the sequencing results. So areas of the red is the host, the human. Areas of the blue is actually the virus part. So from our sequencing work, we were able to find these sequences where they have both the human part and the virus part, and we were able to map this. And we were able to confirm it as well. And this is another integration site where it comes out as a chimeric transcript, having a bit of the human and a bit of the virus coming out at the same time. What perhaps is more interesting is that we were able to pick up the viral integration in complex rearrangement, which may suggest the virus may be first integrate in one half of the part and then causes some sort of disruption of that genome and enhancing the rearrangement. Perhaps even more interesting is that this particular case where the virus actually integrates in one intron, but it comes out with an exon. And that's because there is a cryptic splice site actually within the virus that mimics the host. So it has a new splice site coming out, giving us another chimeric. So we then also, because we have the sequencing data, we can work our way asking the question whether upstream or downstream genes could be affected by viral integration. And based on our work, based on our analysis, it doesn't seem to be the case. The viral integration does not affect whether upstream or downstream 100 kb, which is quite far. So I want to finish with this last story that we have done, which is very interesting and is very close to my heart. And this is trying to understand the inter- and intratumoral heterogeneity. From a biologist, it's very interesting to understand how it evolves and what's in there and all that. But from a clinical standpoint, it's even more interesting. Why? Because if the tumor is heterogeneous as such, that it has too many different components, then it may not be receiving the right treatment, or that may partially explain why some people partially respond and not respond to the full treatment course, whatever. So understanding the clonal heterogeneity is an important information from a clinical standpoint, and it may likely explain many of these therapies that have failed in the treatment of HCC. So in trying to understand the clonal heterogeneity, we have several questions. So the first thing is that very often in HCC, you see multiple lesions. One question is, these two lesions, if it's there in the liver, are they the same? Okay. The second question, is this related but only a subclone of the other one? And then next thing is that if we take a piece of this and we cut it up into different quadrants, are these all different sites of the same mutations? Okay, are there some that is more in one than the others? Or are there some that is shared, or found only in one as what we call a private mutation? So this sort of information is very important for us to understand how the tumor evolved. So this is a case from HKU transplant team, so we're very thankful to them. So the patient has got two nodules, so here one and here another one, and the two tumors actually are separated less than 3 cm. So, so we had the fortune to have this, and we were able to cut it, each one into 4 quadrants. So we call it nodule 1 and nodule 2. So this is the result, the copy number variations from the nodule 1. Looking on top, there are some copy number variations in the few, but not one. Okay? So on this quadrant B, there's hardly any aberrations. But then if we then look at the biallelic fraction, what we're seeing here, there seems to be a gradual increment. This one, most aberrant, and the biallelic fraction, most wide apart, which suggesting it has progressed and much enriched there. So it is, based on this data, it is plausible that the first area, where it kickstarts the whole motion, starts from nodule B, and then D, and then C, and then A. So going this way up. This is nodule 2. If you just look at the copy number variations, there's hardly any if you look at the overall. But the biallelic fraction is telling us it's not the case. There is one consistent copy, what we call the copy number neutral LOH, which means one copy is lost, the other copy is reduplicated, giving us a balanced overall copy. So there is one copy kind of aberration, which is seen in all nodules, and it is likely, it's more homogeneous, coming up at the same time. We were able to also substantiate this our postulation through the somatic mutation analysis, where we see that in nodule 1, the total number of mutations actually varied between nodules, whereas the nodule 2 is more consistent. And we were able to detect also there is a consistent increase in those shared in 2, shared in 3, shared in 4 kind of information based on this. And so there's a gradual increase, and the increase is the same as we postulated from the copy number variation. Here, with the nodule 2, each one of them seems to have very similar mutations. This shows you what is actually in the somatic mutations, what is actually in the tumor. These are the genes that we picked up. Based on the analysis, it would suggest to us that TWIS2 may be a very early mutation that is happening in each of these nodules, possibly TP53 come later on. Whereas in nodule 2, I would say it's PRK3CA, which is a major driver, and also the beta-catenin being a major cause of the carcinogenesis in this nodule 2. HPV integration can harm the body, but at the same time it can be used as a clonal marker, because we know once it integrates and the cell expands, it will expand along with the integration site. So what we saw here is that a couple of integrations in nodule 1, which vary a little bit except the 2 here, and then more actually being shared in the different nodules with the integration. So that again reinforces that this may be following a progressive course. This probably is more homogeneous. And the 2 of them, you can see, is very dissimilar in whether we look at the mutations, the copy number, or the viral integration. The last one is by structural variation. So it just tells us what are the rearrangements that are being picked up in the 2 tumors. So that's what I want to say is that in this one example that I'm giving you, it actually tells us despite the fact that these two tumors are 3 cm apart, and almost invariably you would think it's a metastasis lesion from it, or just a migration out of it. But in fact, they are not. They actually are two nodules arising at the same time. One driven through the TP53, the twist tube, and then gradually progressing, and the other one, very homogeneous, and driven through the beta-catenin, and then the TUG promoter perhaps. And thus, if you were to give drug to this patient, one drug would not work, because it will affect one, but not the other one. You probably will have to give 2 drugs. And this also calls for biopsy of the nodules wherever we see them, and not assuming, just because of the proximity, they are the two of them being the same. So I hope, based on my talk, that I can let you to appreciate understanding the genomics landscape is very important to underpin the molecular basis of the tumor biology. It is really the cornerstone of precision medicine through influencing the targeted therapy. Oops. I'm going to finish with the last 2 slides, or 3 slides, thanking my group. It has been a wonderful 5 years experience working with all of them. We have colleagues from back in CUHK, HKU, and also Hong Kong UST, Shenzhen, and Sun Yat-sen. It has been wonderful working with them. And of course, we have to thank RGC for giving us the resource so that we can be together and then work together for the last 5 years. Within our group, we have actually 3 big teams. So we have the clinicians here on board, and then we have the basic researchers, and we have the genome sequencing and bioinformatics, the very important aspect, that we work sort of very closely together and having regular meetings to exchange ideas. Last but not the least, I think we should be thankful to the patients. We're indebted to them. We are thankful to the patients and to their families for allowing us to work on the tissues. Without those tissues, precious samples, there will be no this kind of research. So on behalf of my team, we thank the patients and their patients. Thank you.
Thank you, Professor Wong. Please remain on the stage. May we now invite Professor Law to come forward to host the Q&A section together with Professor Wong, please.
So let's start the Q&A session. I believe that Professor Nathalie Wong welcomed your question. And any questions from audience?
I have a question, Mary from Chinese University of Hong Kong. It's interesting to see the AA cause a certain signature. And the Chinese medicine, I think the standard text had revised the list of medicine in 2005. So they exclude many of the herbs that have the AA. Now, it will be interesting to extend your finding to look at how long ago the patients were exposed to AA, whether those exposed for chronic interval or acute interval had more likely their signature.
Yeah, so there are 2 aspects here. So with AA, I think people are aware of it, but didn't have the molecular signature, whatever the mutation profile, to actually reinforce the concept. So I think in Hong Kong, I think as you rightly pointed out, quite a few years ago, we have already stopped it, because AA is not only associated with HCC, but also urinary tract cancer and also bladder cancer. So it's just not HCC. But I think in Taiwan, this may not be implemented. So I mean, the government may have implemented, but I think the general public is still consuming this kind of AA herbal remedy. And moreover, AA is actually not found not only in the plant, but also in other plants, which could be mixed as a herbal package. So you may stop the AA, the pure plant, kind of like the consumption, but you probably sometimes can't avoid it in some of the herbal package that is involved. So I think, yes, it calls for some sort of consolidated effort between TCM, to see if they can find a substitution, and also it calls for a good questionnaire for those patients of when they have consumed those herbal remedy and how long ago, and then also a more big data analysis is actually called for in here. Yeah.
Thank you.
So maybe before the second question I follow up this question is that, so it's quite interesting to look at, there's a AA signature in the less related HCC, but I think the HCC, you have a quite obviously precancerous lesion, for example, the cirrhosis in the less patient.
Yeah.
Have you some data to
we're working
sequence the cirrhosis and find the...
So we have the sequencing data of the tumor and tumor adjacent. So yes, we are.
You are looking for?
Yeah,
Okay.
we will be, yeah.
Any other questions?
Very nice talk.
Thank you.
My question is about the origin of the heterogeneity. Do you think each of the different cancer type are derived from same cell type or the different cell type that contribute to the...
Okay, so this is that's literally $6 million question, whether it starts from stem cell or not. I would say yes. It may start from some sort of stem cell aberration and increases the proliferation. But after some time, then the mutations acquired in those subsequent sort of generations of cells actually itself is also quite stemless. So you have so much aberration throughout. It's not only just the stem cell. All the cells that are coming out from the stem cells are highly aberrant, full of mutations, copy number, methylation changes as well. So itself, they will actually have some stem properties that will induce carcinogenesis.
But, like HBV, supposed to infect hepatocyte or also stem cell as well?
It will infect everything, because I don't think HBV, when it integrates into the whole genome, host genome, it's not only in the tumor adjacent will also have, so it's quite random as well.
Questions? So maybe I ask you one more question. So
I should have asked him.
Actually, I think one of the mainstream for current clinical oncologists is they are trying to use the immunotherapy to treat every kind of the cancer. But the most critical problem for the immunotherapy is about the biomarker. So I looked at for some of the liver cancer, they have the hypermutate phenotype, or whether you suggest that according to your study, there's some useful biomarker can help the oncologists for further refine the immunotherapy to treat this cancer.
So there are two questions here. First is biomarker for immunotherapy. Second is the mutation. So let me address the first one. So we are working on a couple of biomarkers to see if they could predict or help with immunotherapy, but we don't have solid data at the moment yet. In terms of mutation, HCC per se is actually a low mutation disease. If you look at the genome, it's about 3 mutations per megabase. So compared to other tumors like lung, colorectal, or melanoma, we are by far low. So what you're looking at, the chart just now of the AA, you're really looking at just HCC per se. You haven't really compared it to other cancer types. In our cases of analyzing it, we have a few of the hypermutated phenotype, but still compared to other cancer types, it's not high.
Well, thank you. Any question from the audience?
We may take the last question from the audience.
I'm still curious about the HBV integration. So any hotspot in the genome?
Yes, we have picked up a hotspot in the genome. So there is the TERT promoter, there are the repeat sequences, and there's also the MLL. So there are a few, but the incidence is, not very high.
Thank you, Professor Wong and Professor Low. Then Professor Wong, please return to the audience, and Professor Low can continue to introduce the next project.